Good morning everyone. So in this video we are going to learn about the phases of the cell cycle. Let's begin. So after finishing this module, the learner should be able to first identify the different stages of the cell cycle. Second, describe the significant changes that take place at each stage. And third, identify the control points of the cell cycle. So let's start with this simple trivia. Just as caterpillar go through a series of stages before running into a turning butterfly, the cells also undergo a certain life cycle. These building blocks of life have to go through growth and development stages called cell cycle. So the cell cycle is uh, the cell cycle or the life cycle of the cell undergoes duplication and growth of cellular contents and division into new daughter cell. So, as human, we also reproduce as well as just like cell, they also reproduce or propagate. Now, the first process or the processes involving the cell cycle is the interphase and the meiosis. So when we say interphase, it is the phase where a cell prepares itself for cell division, and it is subdivided into three stages, namely G1, S, and G2. When we say G1 is GAP1, S stands for synthesis, and G2 stands for GAP2. Then the next process would be the mitosis. Mitosis is the second stage of cell cycle where actual division of cell into two identical daughter cells occurs. So these two are main processes involving the cell cycle. Let's start discussing about the interphase. First is the G1 phase or the GAP1. This phase is the intermediate phase between mitosis and synthesis phase, where a cell is prepared for DNA replication. The cell size and cell components such as proteins undergo replication at this stage before proceeding to S phase or the synthesis phase. So basically, in G1 phase or GAP1, it undergoes or it prepares cell for the DNA replication. Then, the S phase will take over. So, S phase stands for synthesis, and the cell synthesizes an exact copy of its DNA. So, meaning to say, it synthesizes or it undergoes DNA replication. Unlike in G1, it only prepares DNA replication, meaning to say, it prepares all the material inside the nucleus or inside the cell for the synthesis of the organism or of the cell rather. So this phase ensures that the cell undergoing cell division will obtain an identical daughter cells. So that is the uh, synthesis phase where it is it where DNA replication happens. Okay, let's proceed to G2 or the GAP2. When we say GAP2, it is the growth phase for the cell where proteins and other organelles continue to grow and rearrange in preparation for the next stage, which is mitosis. So going back to our interface, in GAP1, it is the intermediate phase where the it prepares the cell for the DNA replication, and also it prepares other proteins which is needed for the synthesis phase, which that is happening in the second stage of interphase. So in synthesis phase, uh, it synthesizes an exact copy of DNA, or it undergoes the DNA replication. So in G2, it is the growth phase where all the necessary proteins, or it finalizes, or it prepares all the necessary proteins and the DNA, of course, which is ready for the next stage, which is mitosis. So in mitosis, we have the so-called, in mitotic phase rather, we there are two processes that happens. We have the mitosis and the cytokinesis. So in mitosis, there are several stages that happen. We have the prophase, metaphase, anaphase, and telophase. And after telophase, cytokinesis happens. When we say cytokinesis, it is where the cytoplasm of the cell divides into new daughter cells during cytokinesis. Okay? So, uh, going back to mitosis, the cell's DNA condenses into chromosome at this phase, and identical set of chromosome is pulled into opposite areas in preparation for division. So, in to generalize, these are the stages of the mitosis. Okay? Then cytokinesis happens after mitosis. Now, this is the stages of mitosis. We have the prophase, metaphase, anaphase, and telophase. In after the telophase, or we can say that after telophase, then cytokinesis happens. So there we can find the separation of two identical daughter cell that uh, that that exhibits the so-called cleavage furrow. So after having the cleavage furrow, then i- the division of the final cell will result into two identical daughter cells. In animal cell, there is the formation of the so-called cell plate, and the cell plate will now serve as the division of the plant cell. Okay? So, the Golgi vesicle develop and fuse along the equatorial plane, creating a new membrane and cell wall for the two daughter cell. So that is the cell plate. Okay? The cell cycle is crucial process which provide vital cells throughout the body. It needs regulatory mechanism to keep from producing cells that are not viable or dangerous to the body, like cancer cells. There are three checkpoints during the cell cycle: the G1 checkpoint, the G2 checkpoint, and the M checkpoint. So in G1 checkpoint, this checkpoint determines whether the cell is ready for the division. The, it checks the DNA damage and the cell size and nutrients. Okay, in the first checkpoint, then it prepares all the necessary proteins, and as well as it prepares the cell for DNA replication. So in G2 checkpoint, it checks the accuracy of S phases DNA replication. So it makes sure that all the DNA is accurately replicated for both in creating two daughter identical cells. So uh, it checks the S phase, or the synthesis, or the DNA replication, before allowing the cell to proceed to mitotic phase. So in mitotic phase, or the M checkpoint, another checkpoint of cell cycle that determines if the sister chromatids are correctly attached to the spindle microtubule, microtubule in the M checkpoint. It checks for misplaced chromosome and posts the mitotic phase to allow proper attachment of the chromosome. So what is the importance of checking point? So the importance of checkpoint is to make sure that all the necessary material for the replication or for the cell cycle is needed before going to the next processes. Because if there is no checkpoint, it might result to cell mutation. It may metastasize and it may result to become a cancer cell. So we have also the regulatory proteins. Okay, so the regulatory proteins are cyclins and cyclins dependent kinase are cell signaling molecules that allows the transition of cells from G1 to S phase to G2 to M. So these are the regulatory proteins or cyclins and cyclins dependence kinase are the proteins that serve as the signal for the cell to proceed to the next process. Okay, then P27 and P53 are protein suppressors that blocks the cell cycle if DNA is damaged. Okay, P27 blocks the cell entry into the S phase and P53 blocks the entry into the M phase. So kapag halimbawa if happens that DNA is damaged, then uh, there is the so-called program cell death na nangyayari. Kapag kasi halimbawa yung proteins na yan ay hindi nag-inhibit doon sa pag-reproduce ng cell, it may cause to the cell mutation or at pwedeng maging ang magiging resulta nito magiging cancer cell yung cell na dapat sana is ma-reproduce into two identical cell. So, uh, regulatory proteins are very necessary in in regulating the cell cycle. So, remember that there are two processes involving the cell cycle. We have the interphase and the M phase. So, there are three processes involved in the interphase. We have the G1, GS, and G2. In M phase or mitotic phase, we have also two processes involved. We have the mitosis, which is separated into four processes. We have the prophase, metaphase, anaphase, and telophase, and cytokinesis that uh, that aids in the cell division or that aids or that supports the cell or that divides the cell into two identical daughter cell. So again, in each of these processes, there are different checkpoints. We have three checkpoints. We have the G1 checkpoint, G2, and the M checkpoint. Okay, so thank you very much for listening and watching in this video. I hope that you have learned something from this discussion. So, if you want to, uh, if you have comments, just comment down and please like and subscribe. God bless everyone.